Never mind. Okay. So this paper, I just titled this slide "Food Allergies." I don't remember what the title of the paper was. I don't really care. But this paper and the whole thrust of this, I think, is the first of what you're going to find many similar studies over the next half decade. I think in many ways, what we're looking at in this paper is the future of how we study and even how we treat food allergies. So, I don't know, the first law of technology, right? We overemphasize the immediate impact and then underemphasize the long-term impact, but maybe I'm falling under that. I don't know. Whatever. So, key terms. Food allergies, which will, from this point, be abbreviated as FAs. And when you read the paper, most of the time it just gives you the abbreviation, introduces food allergies at the beginning. And so in this paper, I think they do a really good job of reminding you or explaining to you that food allergies are genetic illnesses in the same way many other things are complex genetic illnesses. And so I think it does, a really good job of reminding you that if you are making antibodies against things you should not be making antibodies against, like peanuts or dairy or egg whites or whatever it is, if you're making antibodies against that, it's most likely because there's something in your genome that's messing things up a little bit. Now, these food allergies are very obviously complex genetic illnesses in that they are not monogenic, although we did have a monogenic one last time, the favism, right, the allergy to fava beans. But most of these, they're not that way. They are complex genetic illnesses, which means several things. One, we should anticipate there being multiple places in the genome that are associated with that phenotype. Okay? And that phenotype is the production of antibodies against that protein. And because they're complex genetic illnesses, we should anticipate the environment playing a large role in the development of those allergies. And the main place that environment is going to play a role in the development of these allergies is the timing of first exposure. The timing of first exposure. And we saw that even a little bit last week, where we saw a specific mutation in the MHC correlating with the development of seasonal allergies. But if you timed the first exposure to that antigen right, you could overcome having a genetic propensity for developing an allergy against it, right? Remember that? It was awesome. And then I told you about my son and asked you to pray for him. He got sent home Wednesday and Thursday, and now we just have to keep him home until we can get this all figured out. So, anyways, he loves it because he can finish his schoolwork that they sent him home with in like 30 minutes, and then he just gets to play for the rest of the day. So, anyways, sorry. Peanut allergy. So they started this project, and we'll talk more about this in a little bit, with the desire to find places in the genome associated with 9 different food allergies. They basically only were able to find associations with peanut allergy, and throughout the rest of these slides and the rest of the paper, peanut allergy is abbreviated PA. Okay? And anyways, so I think that the title even just references peanut allergy, even though when you read the paper, you're like, they were really trying to find the link with all of these allergies, but all they could do was with the peanuts. All right. HLA gene, and HLA stands for human leukocyte antigen. It is part of the MHC complex. This gene is part of the MHC complex of genes, and so this gene is part of the, or ends up getting transcribed and translated into a protein that is part of the system found in the first responders, the innate immune cells, to present antigen to T helper cells. And so, interestingly enough, the loci that we're going to talk about and focus on, that they found a relationship between the loci and the and peanut allergies. Both of the loci are in these or two different of the HLA genes. Okay, all right, cool. And then this one's my favorite of all the terms: modified quasi-likelihood analyses, abbreviated MQLA. And so throughout the paper, you'll see MQLA, MQLA, and this is what they used for their initial genome-wide genome-wide association study. And it's a statistical tool. It works somewhat like a logistic regression. And so remember, we talked a little bit about this already. When we're doing a linear regression, we need our dependent variable to be what? What do you need your dependent variable to be if you're going to do a linear regression? Continuous. That's right. Right? So, and we'll use it even though we're statistically, it's a little bit suspect, but as long as there's like a minimum value and a maximum value and everything in between is possible, then we'll still use linear regressions, even though technically speaking you shouldn't, unless it is truly continuous, which means any value is possible, right? You're like, with a lot of the things that we use linear regressions for in science, you know, we're talking about, you know, a dependent variable might be human height, okay? And obviously every single value is not possible. You can't have negative heights. Do you agree? Okay, good. You can't even have, you know, a height below some certain value because it's biomechanically impossible. Just like you can't have a height above a certain value because it's biomechanically impossible. But everything in between our minimum and our maximum is possible, and so it's not truly continuous, but we can use a linear regression. But logistic regressions we use when our dependent variable is not continuous, but instead it's discrete. You have certain values that are possible, and so a good example of this might be I don't know, like a widow's peak, where it's yes or no. Or even something more complex than that. I mean, let's just say, for simplicity's sake, for my example, because I want to make this example, and it's not really true, so I'm going to force you to assume that what I'm saying is true. Okay? So assume human eye color was determined by just either a single locus or just a few, and there were basically 4 values. There was blue, green, hazel, and brown, and that was it. Okay? That's it. There's nothing in between. And so then we would still have a discrete dependent variable, and we would still do something like a logistic regression, even though it's more than 2 options, yes or no. Okay? There's 4 options, but they're discrete. It's not a continuous variable. With this modified quasi-likelihood analysis, it works a little bit like a logistic regression, in where your dependent variable basically has 2 options. Yes, peanut allergy, no peanut allergy. Yes, egg white allergy, no egg white allergy, right? So we only have 2 values. But the reason why you can't do a true logistic regression is oftentimes because even our independent variable is not continuous. Because it's basically like you have certain things that are possible. You only have, if it's a single nucleotide polymorphism, you've only got 4 options, A, T, C, or G, right? And then not only that, but it's not every place in the genome that is possible to vary. A lot of the places in the genome are constant. They can't vary. Okay? And so in that situation, not even our independent variable is continuous. It's discrete, and so we need some more kind of sophisticated, creative statistical tools. Okay? And here is one of those statistical tools. But it works a lot like a logistic regression. Basically, our dependent variable is yes peanut allergy, no peanut allergy. Our independent variable, several different genotypes, okay, at several different loci. And you can do it one at a time, or you can do multiple loci together. Lots of ways in which we can do these. Okay? Any questions about these key terms? Okay. All right. So what did they actually do? What did these researchers do? If I don't know, let's just say hypothetically you decided that you were never going to read this paper. But you need to know a little bit about that paper. What do you need to know about what they did? First thing they did was they did a genome-wide association study with 9 common food allergies. And so basically they did all 9 food allergies one at a time and did a genome-wide association study looking for any locus that had a significant ability to predict the presence of that allergy, right? So basically your dependent variable is allergy, yes or no. And then you look at all these different independent variables, and whenever you find one that has a significant ability to predict yes versus no, now we've got an association, right? And then we can use that to figure out what's our probability of making a type 1 error, basically what is our p-value, and go from there. So that's the first thing they did. Then what they did was they replicated their study using a very specific cohort of people. And for this one, they did it only on peanut allergy, and that's because their genome-wide association study only found a relationship between their single nucleotide polymorphisms that they used and peanut allergy. No other of the 9 allergies that they used had any loci that provided a significant ability to predict the presence of that allergy. They only looked at about 600,000 single nucleotide polymorphisms, and so being that that's 600,000, what fraction of 54 million is that? I don't even know. A little bit more than 1%. Is my math right? Yeah, it's a little bit more than 1%. And so that's not to say that there's no genetic basis for these other food allergies. They didn't search wide enough. Okay, their net wasn't big enough. You know, it's like you got a little net next to the boat and you swoop it in. You're like, oh, there's no fish in this lake. That would be an absurd conclusion to make, right? Sorry. Thank you so much. Thank you, Kaylin. And then the last thing that they did was after they repeated this to basically confirm the relationship between the specific loci and peanut allergy, they looked at different methylation patterns in people that had the genotype that would typically result in a peanut allergy but don't have a peanut allergy. They looked at the epigenetics and to determine if you have a genotype that should result in peanut allergy but you don't have the peanut allergy, is that because you methylated the version of that gene that is mutant, that should actually result in a peanut allergy? Because they wanted to look at that as well. Really cool. A really cool thing that I think they did that took this from being something that would have been published in a really good journal to taking this and making it something that was published in the best journal on earth. So, you're like, this is Nature Communications, not even real nature, Dr. Engel. Relax. Still has an impact factor of like 30. Which a good journal has an impact factor of about 1. Which basically means if it makes it into Nature, everybody reads it, knows about it. Yeah?
What do they define as the peanut allergy? Is that like someone said they had a peanut allergy? Did they take like blood tests?
That's a good question. I don't know. I don't recall having a lot of information about when they did that second study and they or even the initial study, what they used to determine whether or not somebody truly had a peanut allergy. My guess would be is that they used only people that had a confirmed response to it. And so either you could see a confirmed response to it, or they took a blood test and confirmed that they are making antibodies against that protein. But I don't think they give any information about that in the paper. Yeah, I don't remember reading any of that. It's a good question. Okay, so this is what they did. So let's just say, again, you're like, I'm never going to read this paper, and but you're asked a question on what did they do. Here it is. Here's your information. You're welcome. Now the next question: what did they find? What did they find? And so what they found was that they found approximately 40 loci in the HLA, human leukocyte antigen, the DR gene, and the HLA-DQ gene. I almost said DG, but that is not a G, that's a Q. On the P arm of chromosome 6 correlate significantly with peanut allergy. So they found about 40 loci in these regions on chromosome 6 that correlate significantly with peanut allergy. Now that's a lot of loci. That's a lot of loci. And so here are the two that had the most significant relationship with peanut allergy. And so these are the names of the single nucleotide polymorphisms. Here's RS7192 and RS9275596. Great names for these single nucleotide polymorphisms. But if you have 54 million, I mean, what else are you going to do? You kind of just have to name them with a number. Like, you can't give them a personal name. Plus, they're not people, so who cares? But if they had nice creative names, it might be easier to remember. But so the probability of making a type 1 error, and in this context, a type 1 error would be saying this locus correlates with peanut allergy when it really doesn't. The probability of making that type 1 error, 5.5 times 10 to the minus 8. It's a really low probability of making a type 1 error. We are very confident that there's a relationship between this locus and peanut allergies. Here, even better. 6.8 times 10 to the minus 10. Very low probability of making a type 1 error, so we are extremely confident that this locus influences the development of a peanut allergy. And then what they found is the remaining loci, about 38 of them, are physically linked to these single nucleotide polymorphisms. So they don't actually correlate with peanut allergy, they just happen to be linked with loci that do. Do you remember the MHC thing? I used that example a little bit earlier in class, and then we talked about it on Monday, maybe even. Probably wasn't on Wednesday, where it's a section of the MHC and it correlates with a propensity towards like developing an emotional addiction to smoking. And it's really not because the section in the MHC, but it's because that section is physically linked to a gene that if you have a certain mutation, you enjoy the smell of cigarette smoke. And so you become significantly more likely to develop an emotional addiction to it. Any of you like the smell of cigarette smoke? I love it. I don't know what it is. I must have that mutation because everybody thinks I'm a freak when I tell them that. So that's fine if y'all want to think that I'm a freak. I love it. I don't know what it is. I love it. I don't smoke cigarettes, but I love being around people that do. But I have to limit my exposure because I understand it's not great for me. But every now and again, it's just, I don't know. Can't help it. It's in my genetics, you know? So these two important single nucleotide polymorphisms, they did some additional studies, which I actually didn't mention in the previous slide, but it's relevant here, to demonstrate that these two single nucleotide polymorphisms play a role in the methylation of other places in the genome. So these two single nucleotide polymorphisms that correlate significantly with the development of a peanut allergy function in the methylation of other parts of the genome. Meaning they play a regulatory role. You're like, that's fantastic, because you have these single nucleotide polymorphisms are in genes. Genes that code for some of the proteins of the major histocompatibility complex, right? Those proteins that present antigen, foreign material, to T helper cells. So these loci are in genes and yet also play a regulatory role. Wow, that's really interesting. And so it's not even as simple as what we've already found out about the genome, that less than 2% of it gets translated into protein. About 80% of it gets transcribed at some point. Now we have evidence that there are regions that get translated but still play a regulatory role somewhere else. So maybe they'll get translated sometimes, but they might also be transcribed into regulatory RNAs. Like, wow, this is really weird. But anyways, that's another story for another time. Now, something they did with the last thing that the last piece of work where they studied basically, okay, some people have this genotype but don't develop peanut allergy. We hypothesized that they only have one mutant copy and it's methylated, so it's not being expressed. And this is what they found. That the mutations in these single nucleotide polymorphisms, they can be overcome by the genome being methylated in that region where that single nucleotide polymorphism is. So you could be genetically predisposed to develop a peanut allergy, never develop a peanut allergy because you only have one copy of the mutation that leads to the development of a peanut allergy, and it's shut down. And only the normal allele is being expressed. You're like, fortuitous. Yeah, Caleb.
How would you shut that down? Like, is there something you could do to...
That's a wonderful question. The answer is yes. We can methylate sections of the genome in a very focused way. The problem is you'd have to do it really early on in development so that methylation pattern was inherited through all those mitotic divisions. Once you get to a point where you're like 6 or 7 years old and you go into anaphylactic shock from eating peanuts, right, and they resuscitate you and you're like, man, what are we going to do? It's kind of hard to go and take and genetically engineer the trillions of cells that make up your body. Can we do it? Sure. It's going to be hard and really expensive. Be a lot easier to just be like, hey, let's change your environment. Let's avoid peanuts. You know? If you don't know whether it has peanuts or not, don't eat it. Don't eat anything at people's houses. Sorry. You're going to be the weird kid that's like, I can't eat it. I'm sorry, I have an allergy. Although I think peanut allergy, it's like up to almost 5%. 5% of people have a peanut allergy somewhere. You know, not everybody has a severe peanut allergy. So, you know, there's 1 in 20 other people that are freaks with you. So if you're at a party of 40 people, there are probably 2 of you. So, maybe.
You would probably only have to methylate the immune cells, right? Because then they're the
Maybe.
ones that make you go into anaphylactic shock.
Yeah, the problem with those is they're not long-lived cells. So every time you get a new round, so you'd have to methylate your hematopoietic stem cells. You'd have to, modify the hematopoietic stem cells. So then every round of blood cells that they produce will, that's doable. I mean, so now we're talking about millions of cells versus trillions. Maybe tens of millions. I don't know how many hematopoietic stem cells there are. There are a lot because the same stem cells result in all red blood cells and all white blood cells. And I don't know if you know this or not, but a red blood cell only lasts about 30 days. So you make a lot of them. So they're probably billions then of hematopoietic stem cells would be my guess. Hundreds of millions or billions, but that's less than trillions. Markedly so. David.
How does like some people can build up a toleration to peanuts by like microdosing?
It depends on the nature of your allergy, right? Yeah, it depends on the mechanism of why you're allergic. If you're allergic because you're making antibodies, it's a little bit more complicated. If you're allergic because your like macrophages are presenting that antigen to T helper cells and eliciting some response, it's a little bit simpler to kind of develop a tolerance. Yeah, but yeah, well, I mean, we already know that the development of an allergy is very time dependent. The time of first exposure plays a very large role in whether or not you develop the allergy. And so the American Society of Pediatricians, what's that organization? Is it that? American Society of Pediatricians? I don't know. I almost said parasitologist. That's my organization. I'm a voting member, you know. Anyways, they recommend not giving your kid peanuts until after the age of 2, because if you exposed to peanuts prior to the age of 2, you're more likely to develop an allergy to peanuts.
How about during pregnancy?
What's that?
How about during pregnancy? Or some people say don't eat peanuts when you're pregnant?
Yeah, I mean it's the same thing. Like it's just the antigen from the food you eat can move transplacentally. Yeah, absolutely. So last thing, well, what can we do? What do we do with this information? What do we do with what they found, and how do we use it? Well, one, it allows us to predict the presence and the severity of a peanut allergy. So knowing that this could be a very severe issue, a very severe genetic illness, might this become part of the screening you do at birth, right? In the same way you can screen for other things, might this just be something you screen for at birth, right? You look for just these two specific single nucleotide polymorphisms. When you take that little blood sample from the heel of the infant and you check for various things, use some of that to figure out what alleles they have at those two loci. And if they have a propensity to develop a peanut allergy, you keep them from peanuts, right? Change their environment. Also helps us determine the best method, because once we know where in the genome you have an association with this genetic illness, we have a better idea of what cells we target, like what you were pointing out, Nathan, and how we target those cells. And then the last thing we do is we realize, man, this is a really good paper, probably the reason why I made it into Nature, and we take something that they did, we expand it a little bit, look at some other single nucleotide polymorphisms, and find correlations with other food allergies. Okay? And so we can do a similar thing, maybe make it a very simple routine screening. Because if you've got two loci that play a really important role, and then you have, a handful of loci for this food allergy and this food allergy, and you've got, maybe 10 fairly common food allergies, and it's like 50 total single nucleotide polymorphisms, that's a very inexpensive screening procedure, right? I mean, you could do it for less than $10. I'm pretty sure insurance companies would pay for that. Versus, if it allowed them to significantly reduce the risk of having to pay an enormous bill for a kid that goes into anaphylactic shock for eating something that they're allergic to. Yeah. Do y'all know Mita Kate? Yeah. Did you ever hear a story about the last time she went into anaphylactic shock over a peanut allergy? So hers has gotten worse and worse the older she's gotten, and the last time it took like 8 EpiPens, and it was almost to the point where they had to take a needle right into the heart with, adrenaline. It's just not good. Yeah.
Where did they get the DNA they used for this test? Did they get this from serum?
You're talking about in this or just in general?
In this.
I don't think they said.
Because there could have been a difference between the blood cells that came from the hematopoietic stem cells and then just regular somatic cells.
Sure. My guess would be is that they would do it the general way that you would do anyway, and where you use saliva. Yeah. But I don't think they ever said that either. I don't remember reading anything about it.
They don't have a very long methods on this at all.
No. No, just their statistical tools is really what they describe. Mm-hmm. Yeah. I'm sorry. Okay. Questions? You just want a demo of service?
Sure.
Okay. Let's do it. Oh boy. Okay. Okay. All right. First thing you need to do is you need to take the text files that you have, right? You have text files that basically go through all of these, and you need to convert them into an Excel file like this, where your first column is your individual, and you're only going to have, you can put all 3 of your individuals in here, right? You can put, like if you're using my genome, you can put Dr. Engel here, then hypothesis 1 individual here, hypothesis 2 individual here, right? So you could have all 3 in the same one. And then, oh wow, you can't even see. It cut off part of the image. Oh, you lost the first 2 columns. What do I do? Okay, anyways, it doesn't matter. Over here, it just says individual and then has the names of the individual, and then here it has sex, and that's because this is a stock file that they produce, and this is about parental analysis. But we're using this a little bit differently. We're not going to do a parental analysis because it's a little weird. But notice, for all of these loci, they basically have 2 copies of the locus. So FC8304 copy A, FC8, oh, that's B, that's not an 8. FCB304B, the second copy of it, and then they write the two alleles. Now, this program is designed to work best with microsatellites. And so these alleles are the number of repeats of whatever that microsatellite region is. And so if you want to simplify this and get it to work more quickly, because when you use single nucleotide polymorphisms and you use letters instead of numbers, it takes a little bit more time to run the system. So what I would suggest is that you use just numbers for A, T, C, and G, right? Where you say like, and then just have a key, right? Where you could like, if we don't know what it is, because you'll see that in the notepad here, let me show you one of these pieces of data. So you see at some of these locations, we don't know what it is. So I would represent that as a 0, okay? And then just use 1, 2, 3, and 4 to represent A, C, G, and T alphabetically. That becomes very simple, right? A is 1, C is 2, G is 3, T is 4, right? And then you can just go in and the nice thing about this is if you just copy this, and I'll show you how to copy the data from here into Excel, you can go in and do command find and replace, and you can have it replace all of the A's with a 1. And so you don't have to go in and do it manually. I'll show you how to do that. You're gonna like it. You're gonna like it a lot. Okay? So that's the first thing you need to do is you need to make your data look like this, and I'm gonna show you how to do that. Is that what you wanna see?
Yeah, well, just because there's a ton of numbers on
Oh, that's why actually the first thing you need to do, but this is something we already talked about, is you need to go in and find the target loci. So you need to pull some papers where you know that there's certain regions of the genome that correlate with what you're looking for, right? So if you're testing like ancestry, not every single nucleotide polymorphism is important for determining ancestry. You can find papers that'll be really nice for writing your reports and will help you to find a target region. Okay, so let me go to this. I'm gonna have to move all of this to the middle so you can see it. Okay, so here's your data file, the one I put together that will allow everybody to assess their ancestry work. And so we can go and do this, and we're just gonna copy it. We're gonna copy. I don't know. We'll take. Let's say you're like, oh yeah, the very beginning of chromosome 1, it correlates with it. It's cool. And so we'll take that and we'll copy it. And then we'll come here and I want an entirely new Excel file. Oh, look at that, that's so beautiful. And then we're just gonna paste it. And look, even though it was kind of wonky and some of them like that had shorter names ended up moving over, when it pastes, it's all beautiful. You see that? It all pasted into nice columns and we're like, cool, here's one locus, here's another locus, here's another locus, cool, cool, cool. And then right now we're going to do this find and replace. So let's see, Control F. Okay, you see that? This comes up when you hit Control F, which is our find command, but we don't want to just find it, we want to replace it. And so we're going to replace any A with a 1. And we're going to say find all. Oh, we're going to replace all. Sorry, I don't want to find all, I don't care where they are. We're going to replace all. Okay, 29 replacements, cool, whoop-de-doo, look. And now all of our A's have been replaced by a number, and you didn't have to go in and manually do it, right? And we're like, okay, well that's cool, that's only the A's, Dr. Engel, let's do it again. So Control F, it's our find command, then we're gonna replace, and we're gonna replace all of our C's, we're gonna replace them with 2's, and we're gonna replace all. Oh, yay! And then we're gonna do that for our G's. And you don't even need to capitalize it. I'm capitalizing it because I'm silly, but you don't need to. If you put in a lowercase g, it'll find the capital G as well. Oh wow, there are a lot of G's. Wow. Wow. T's, here, I'll do that one lowercase just so you can see. Oh, 30 replacements. Cool, and now we've done it for all of them, and now we have no more A, T, C's, and G's. They're all numbers. That's what we want, right? And we're like, cool, but Dr. Engel, the file you showed us, the loci weren't like this, they were like this, right? Do you remember that, how that was structured? So we need to change something. So we're gonna highlight all this. Look at this, we're gonna highlight it all. And we don't want it to stay there because I don't want to just copy it and paste it. I want it to disappear. So I'm gonna go, I'm gonna highlight all of these. Instead of hitting Control C, which is copy, I'm gonna hit Control X, which is cut and paste, right? And so now it's gonna disappear as soon as I paste it. Then I'm gonna go here and I want to paste it, but I want my options. Paste, why is it not giving me my paste options? Oh, will it not do it if I cut it? Oh, so now I'm just gonna copy it. Oh, there we go, now we have paste options. It didn't like that cut. You have to go and erase it later. So I copied it, and then I go to paste, and it gives me all these options, and I just want to do the transpose one. So now instead of copying it in a column, it copies it in a single row. Right? And so now it's in the good format, and you're like, cool, it did exactly what I want. Oh, wait, I didn't click it. And then you click it, and it does exactly what you want, but you were like, but wait, Dr. Engel, the file you showed us had 2 columns for each locus, right? But here's the beauty of it. The program doesn't care whether the 2 columns for the same locus are side by side or whether they're separated by a bunch of things, as long as they're titled correctly. And then so you just go in here, and at the end of all of these you add a little letter A, right? Which will take some time, and that's why I told you it's like a 30-minute of prep time, right? I told you that the prep time's gonna be 30 minutes, because you're like, Dr. Engel, I could do this super fast. You gotta go in and add an A next to all those, so that'll take some time. And then we're gonna scroll all the way to the end here. Look at this. Well, oh, I went too far. Oh, then we'll go back there, and we'll paste it again. Transpose, and now we have 2 copies of every locus, and after you do the second one, you'll go in and you'll add a little letter B after all of them. And now it's set up exactly like how that file was, right? Right? Yep. Except for you'll have to go here, and then I want to cut this. No, it's not going to work. I want to delete this. Yes. Okay, but then you need to come here, and you actually need to type individual, because the program reads that title. Okay, so you're going to type individual, and then down here you'd put Dr. Engel, and then you'd put hypothesis 1, hypothesis 2 if you want to do that, although you're going to need to use different loci for your hypothesis 2, so you might as well just do it as a completely separate file. But anyway, it's up to you. Unless you find some loci that correlate with both ancestry and whatever genetic illness, then it's like 2 for 1, right? That's a wonderful thing. And then you'll have to go in and you actually have to type the loci. So then you'll go in and you can see at that first copy of this locus right here. So this is our first locus, right? Which now is pasted over here, and it's got a 4 there. 4, right? Right? And so unless you know, if there's a trick, and if any of you know a trick where you can take this binomial data and split it between 2 cells, because if you do a split, there's a split command, and sometimes it works. Here, let's see if that actually works, because if that does, then you could just copy and paste all of these and it would be way faster. Format? No. Where's my split command? Somebody tell me. There's my merge command. I don't want merge. I want a split. Right click. Where's my split? Format cells. No, it's not there. Wait a second. I'm going to try this. I'm going to copy this. I'm going to put it in a Word document. Okay. Now we'll highlight this whole thing. And now we'll split it here. Design, layout, split cells. Number of columns, 2. Please. Oh, no, it didn't do it. It didn't split the individuals. No, so it's not going to help us. Get rid of this. Okay. Don't save. So you'll have to go in and you'll have to just look at this section here and just pop them in. And pop them in, right? That's why I told you prep time might be 30 minutes. Because if you have got, 200 loci, it might take a little while to copy those. Because the problem is it's giving us both copies of it, and you need those 2 copies to be separate, right? So you can't just copy it and paste it all in there. I guess you could. and then you just erase the second one. That would save you the time of having to manually copy both. You'd only have to manually copy one. You'd paste one, and then you'd just erase the, but that might take you just as much time as copying it, erasing the second. Ooh, I wonder if we could do this. Oh my gosh, we totally can. Check this out. Watch this, watch. We can format cell. Where's this? Oh my, I clicked the wrong thing. We can format cell, sorry, I'm just trying to save you time. We can format this, and we'll say it's a number, and we only want a single digit. Oh, but that's decimal places. Can I round it? Because we can reduce the number of decimal places, but can I get it to just, because if you could get it to just do it to, you know what we could do? You'd only have to worry about the ones that are heterozygous. Check this out. Because you could have it find, and instead of putting in replace A, you could have it replace AA with that letter, and then so it would only have a single letter, and you could paste all those single letters twice, and you'd only have to worry about those that were heterozygous to do the second one. That's just to save you some time. Anyways, this is how you would build your file. I'm just thinking about ways to save you some time so you don't have to manually. That's where it's getting really complicated and really confusing. I'm sorry, I feel like I've messed everything up. But now this is set up like that file is once you have all your data in, right? It'd be set up like that Excel file was. Yeah.
When clicking on like data and then like on the tab for data and then the text to columns
Data,
right there. And then, but you have to like.
but they're not separated at all. So delineate
Like, yeah, you have to have a space.
a comma or a space or a tab.
And can you do like a space, like replace all A's with spaces or something? Like a and then add a space and then you automatically put the space next to it.
You could. I mean, you can have it replace it with anything you want. That's actually a great idea. Wait, no, cancel this. So when you're doing your initial replace, you could have it replace just basically all of your combinations. Like say it's AA, you could have it replace it with 1 space 1. And you can have it find AG and replace it with 1 space 3, right? And so it's a little bit of work at that front end, but then watch when well here, there's no more AAs. So now let me have it find 11 and replace it with 1 space 1. And now it did it. Now there's a space in between them, and now we can go in, oh, better yet, let's do this. Sorry. 1 space 1, and I'm gonna replace it with 1 comma 1, just so that there's no confusion that it's 2 separate pieces of data, and then I'll replace all. Oh, you see? Now they're 1 comma 1. Oh man, this is working nicely. And then we'll go here, and we'll go data, and we'll go text to columns, and delimited, and we're gonna go next, and it's comma that separates them. Oh my gosh, it's splitting them! Next, and we're gonna finish. Oh, it split them! It's gonna work! Mia, you're a genius! Prep time may have decreased to 23 minutes. Because then once you have them split, then you can just go through and you copy the whole thing, right? And we copy the whole thing, and then we go and we paste it with the transposed, and then it'll put them all once they're split. Oh, Mia, you're a magician. But then once you have it set up to where it's just like the other file, then you can open up service, and we're gonna go to service. Oh man, I gotta make this smaller so you can see the whole thing. Okay. And then we're gonna go to analysis, access.
Okay, so far.
If it seriously won't open this program, I was playing with this program all morning. Let's try this again. And right now this is being recorded, by the way, so all of the madness you get to, oh, there we go. All of the madness you get to experience again. So the first thing we're gonna do is we've got to have it create an allele frequency file for us. So we're gonna go through, you've already created your genotype file. That's what we were doing in that Excel file. Oh, and by the way, when you go to save that, save it as a CSV. Don't save it as an Excel workbook. Just click Save As instead of just hitting Command S. Go to Save As and save it as a CSV. And then so we're gonna find our genotype file, and I've got these demo files that it comes with. Source data, and here's our genotype. And see, it's there as a CSV, which is what you're gonna save yours as. You created that Excel file, and it's got a header row, and read locus names, the ID. Oh, make that first column ID, but you'll tell it where it is anyway, so then it doesn't need to matter. It's just, we have to make sure we have header row, because our header row is naming the loci, and then whatever you named your individuals. And so you tell it where those individuals are, which they're gonna be in your first column where you wrote Dr. Engel, doctor or hypothesis 1. Your first allele, ours was in column 2 because we didn't have anything else. We went straight from ID into loci, and then our total number of loci. And in this file, there are 9. In your file, there are however many you put in, right? If you put in 200, then you put 200 there. And then it just wants to know where you want to save it. And we're going to save it here as awesome. Oh, I left off the O. I left off the S. The O was there. And we're going to click Save. And now we're good. And then the rest of this you don't really care about. You can have it not do this or do this. It doesn't really matter. And then we're going to click OK. Oh, 9 loci, but 10 names were found. Okay, fine. So we'll go back in here. There are 10, not 9. Boom. And it ran this file for us, and it saved that file. And that's what you need. You need that allele frequency. And how long did it take to run that? That is just, this is a bake time is very short. These are no bake cookies, right? It's all prep time. And then we're gonna go through, so you had to run that first analysis, your allele frequencies, then you'll go and you'll run your identity analysis. And now what it's going to do is it's basically gonna take every individual in your file and it's gonna see if they match according to your rules. According to your rules. So you'll go through, you'll use that same genotype file that you already created. We've got a header row, the individuals are in 1, the first alleles in comma 2. Testing sex separately, we don't need to do that. Allele frequency data, look, it already put in my awesome, the one that you created. And then it wants to know where we're gonna save this, and we're gonna save it here as super awesome. By the way, you don't have to use these names. And then it did that for this and for our identity data. And then this is where you set your guidelines. Minimum number of matching loci. If you hypothesized that Dr. Engel was 50% Irish or whatever, then it should match at however many half of your loci are, right? So if you put 200, here we only have 10, so let's say it's gonna match at 5 at half of those. And then we'll allow fuzzy matching, so this will allow you to match even if you have sections that don't match, and we'll allow it to have 5 mismatches, okay? Right, we wanna know, is it matching half of those key loci? Do you understand that? So we're like, we need to find minimum number of matching is 5, allow for 5 mismatches, and then boom, we'll run it. And it'll tell us the number of genotypes that were found to match. You only have 2 individuals, right? You have Dr. Engel, or whomever's DNA you chose, and hypothesis 1. And then in your other analysis, you'll have Dr. Engel and hypothesis 2. You only have 2 individuals, so if this comes back 0, guess what? Whatever you hypothesized isn't true. If it comes back 1, guess what? Whatever you hypothesized is true, right? Make sense? Does this work? This is way better than just recording a video for you, because we got to do this live. Mia gave us a trick to actually separate those data and cut down prep time. All right, any questions? Do y'all feel like you could do this? And even if you don't, you can go back through this video where it walks you through the steps.